So it's a great pleasure to be here. And I'd like to start with a simple question. So what is this number? Huh? It's a large number, yes. Correct? Huh? Number of insects, almost. Huh? No, no. Number of ants, exactly. It's almost like the number of insects, because you know, once you have the ants, you have everything most. We know it started 100 million years ago. We heard this morning, and they are everywhere since then. So it's a large number. It's not easy to see what it means, but I think there's a simple way to see what it means. If you take all ants, they're small. You take all of them, put them one in front of the other one, you can go 165 times to the sun and back. So that gives you an idea about the number of ants in the world. There is only one other organism which can compete with the ants. It's a simple organism, creates a lot of problems. Huh? Bacteria? Or it's not an organism. Okay, no, it's this one. So humans, so if you weigh all animals on Earth, you will see that humans and ants are about the same. So humans reached the level of ants last week. So now the biomass of humans and ants is the same on Earth. So ants are quite important, and they are interesting because they are interesting to study polymorphism. So if you think of polymorphism, you can have variation which is due to genetics. It can be the environment. You can produce different morphs from a single genotype. It can be linked to the sex. So here you have a nice one, which is a male and then a female. And also in humans, you can have variation in phenotypes. So you can have two individuals which look very different, but inside of the head, it's very similar. The brain is... And it's hard to believe, but this is the same species, basically. At least it's still classified as a single species. And ants are interesting because you can have a lot of variation in phenotypes. In a single colony, you can have the queen, which is very different from workers, but workers can also differ a lot in size and also other type of polymorphism. And another type of interesting thing is that in many social insects, you have variation in social organization, with colonies which have one or multiple queens, and this is associated with many other changes, as we will see. So the caste system is amazing. In a colony, you can have all those workers, the male and the queen. Here you have a worker sitting on the head of its sister. And so this is used to be thought that it is a phenotypic plasticity. And those different castes are associated with many adaptations. And one of them is the queen and the workers. So it's a bit like in humans. There's one group which does all the hard work, the workers, and this one doesn't do much. He lays the eggs, and that's about it. And this can be associated with many adaptations. One of them is the lifespan. So insects don't live very long. So here are different groups of insects. Typically, they live a few weeks. But there are three exceptions: the termites, the bees, and the ants, which can live a very long time, up to 30 years for some ant queens, which is really amazing for an insect. And on this phylogeny, one can see that the evolution of social life has been accompanied by the 100-time increase in the lifespan of individuals compared to the solitary insects. And also so that will be like if you would find a primate which lives 4,000 years. And what's also quite amazing is that the workers, which have the same genome as the queens, have a much shorter lifespan. So it's a good system to see what differences in gene expression underlie variation in lifespan. So the question is how to become the nice person on the throne, the queen, or a worker. So it used to be thought that it's purely environmental. So in ants and social Hymenoptera, males develop from unfertilized eggs, so they are haploid. And females can develop either into queens or worker depending on the food which is provided to them and some hormones. And so textbooks say there's no genetic difference, but as you should know, you should never trust textbooks because there are many things which are correct, but there are so many things which are wrong. So in the last few years, we found several cases of unusual type of reproduction in social insects. So in that species, which live in the southern part of the US, you have two genetic lineages, and queens mate with males of the same lineage, and all the offspring develop into queens, but they also mate with males of another lineage, and all their offspring develop into workers. So in the population, you have queens of lineage 1 or queens of lineage 2. Both of them mate with male of their own lineage and males of the other lineage. And all the offspring which are fathered by males of the other lineage develop into workers, which are type of hybrids between lineages. And so you have pure lineages which are transmitted through the queens and through the males. Then we found another species which is in southern France and Spain, where queens reproduce both. They use both parthenogenesis to produce new queens and sexual reproduction to produce workers. So this is good because sex is nice, as you know, but it's also costly because it means, well, you can get a disease, but it's also costly because you transmit only half your genes to your offspring. So here, these queens have found a way out of that. They maintain sex to maintain genetic diversity in the workers in the colony, but they reproduce by parthenogenesis to transmit their genes to the new queens. And then we found another species, which was quite the same, but here it's clonal reproduction. So all queens are produced clonally from the mother. All workers are produced sexually. In that species, workers are completely sterile. So this is really bad for some individuals in that population. Which individual? Is there a male in the group here? So for males, it's really bad because it means all their offspring will develop into workers which are completely sterile, and normally males are produced from unfertilized eggs. So if all queens are produced clonally from the mother, it means males will have a zero reproductive success. But males have found a way out of that dilemma. What do they do? A young male here? Any idea? An older male? They also reproduce clonally, which looks strange because the sperm is not very efficient alone in the nature. But so what can they do? So the sperm enters the egg. It removes the maternal genome. Then you get a new haploid individual, which is a clone of its father. So in that species, all males are produced clonally from the father. All queens are produced clonally from the mother. And so there is no more gene flow between males and females. Their genomes come together only into workers which are type of hybrids. And if you put them on a phylogeny, queens, females are all on one part of the tree, and males are all on another part of the tree. So they've been the first cases, but now there have been quite a few similar cases which have been reported in other ant species. And probably 20% of ant species may have this type of unusual mode of reproduction. So now I will move to the second part of the talk, which is variation in social organization. And I'd like to thank two of my students and a colleague, Ken Ross, with whom I've been working many years. And so I will report some work on the Solenopsis invicta, which is a species which is native from South America. It has been introduced in the US, and it has a slightly painful sting. We thought it could be a way to control Americans, but it's apparently not strong enough. But we can try to engineer even more efficient ants. And that species, it's interesting because you have two social forms. A form with single queen. As I will show, they are large. They use independent colony founding. That is, the queens depart on mating flight. She mates with one male and start a new colony on their own. And that form, there is strong aggression between colonies. And there's another form in which you can have up to 100 queens in the same nest. They are smaller. And instead of flying away and starting a new colony on their own, after mating, they return to an established colony. And in that social form, there is no aggression between colonies. That is, workers and queens can readily move between colonies. So when I did my postdoc in Georgia, we were interested to see if there was a genetic difference between the two social forms. So we were using allozymes, so that was old time. And here are, for different allozymes, the most common allele. And as you can see, for most loci, there was no difference in allele frequency. Since then, we have used other markers, microstylites, mitochondrial DNA, and we show that there is extensive gene flow between the two social forms. But there was something special for two loci, GP9, which means general protein 9. We didn't know what it was at this time, and phosphoglucose method 3. And for those two, there was important variation in allele frequency between the two social forms. So I will present you data for this locus, but explain you why this one is so different. So we did many experiments to see what was happening at those loci, and we will try to summarize that in a single slide. So we found that in the monogiant form, everybody has the same allele. So at this locus, GP9, you have two alleles, the big B and the small B allele. And in the monogynous form, everybody is a homozygote for the big B allele. So those colonies, they produce new queens, the winged queens, which depart on a mating flight. They mate with one male, and they can fly up to 10 kilometers to start a new colony on their own. In the polygiant social form, things are more complex because you can find other genotypes. You can actually, when they close young individuals, you can find all three types of genotypes with a small B allele. So you have homozygote for the big B allele, or heterozygote, and individuals which are homozygote for the small B allele. And we did experiments to see if those genotypic differences are associated with a difference in phenotype. And we found that there is indeed important variation with size differences and fecundity differences. The small b individuals being much smaller than heterozygotes, which are much smaller than big B individuals. And it's also associated with fecundity differences, this one being more fecund than the two other genotypes. And then we did a series of experiments to see what was happening. And also, very importantly, we found that the reproductive queens are all heterozygotes. In the colony, you never find homozygote queens. You never find queens which are homozygote for the small B allele. So that suggests that there is some selection happening, and this is why we look to those phenotypes. We look for phenotypic differences. So we did different type of experiments, and we found that the small b allele behave like a recessive lethal allele, so that all females, after they close from pupae, when they have the final body size, during the 2 weeks after eclosion, they will die. So we have an explanation why small b queens do not occur in those colonies. It is because they died during those two weeks after they close from the pupae. And then those individuals, actually, they are much smaller than those ones. They don't accumulate fat after they close from the pupae. And so they're unable to start new colonies because during colony founding, the queens feed the first larvae by histolysing the allozyme muscles and using the fat that she accumulates during the 2 weeks after they close from the pupae. And those queens don't have enough fat, so they're unable to feed the first larvae, so they cannot start new colonies on their own. But they will be readily accepted in established colonies, and so there is one way for them to survive. And those queens actually are the same size as the one producing monogiant queens. They can start new colonies on their own, but they will never be accepted in polygynous colonies. And we did some experiments, and we found that there is, the behavior is associated with the genotype, and this is because the workers which have one copy of the small b allele, they will kill those queens when they start reproducing. So it's a type of selfish gene which induces the workers which do carry that gene to kill all the queens which do not carry one copy of the small b allele, with the effect that those queens can never be reaccepted in the mother colonies. And so this is why you only find heterozygote queens. And we have genotyped thousands of queens and never found homozygote for the small b allele or for the big B allele in those colonies. So when we did those experiments, that was a long time ago, we didn't know what was GP9, but the gene has been cloned and sequenced by Michael Krieger, who was one of my students and worked with Ken Ross. And they found that it's an odor-binding protein, which in insects can carry odor in the hemolymph, in the blood of insects. So it could be that this gene is responsible for odor differences between queens. And we know that the workers, they recognize queens of the 3 genotypes. Those heterozygote, when they kill those queens, we know that they recognize the 3 genotypes based on odor differences on the cuticle. We did experiments, and we showed that there was those odors which elicited the behavior of heterozygote queens to kill those queens. So it could be that this OBP affects the order of the queen, and it could also affect the behavior of workers of different genotype. But so we showed basically that a single genetic element, which is GP9, or something which is associated to this OBP, influences the behavior of workers. It's responsible for the presence of two social forms, the monogene and polygene form. And that was interesting because that was the first genetic element shown to influence social organization in any type of organism. And there are 5 other closely related species which have also the monogene and polygene form. And we know that it's also completely associated with the genotype at GP9. So the same genetic element influences social organization in 6 ant species. So the question is, can a single gene, this order and binding protein, account for the many differences, the queen differences in size, the mode of colony founding, the lethal effect, we showed also there are different worker size, worker behavior, sperm content of males, or are there other genes which are linked to GP9? That is that a supergene? So my students many years ago suggested we should sequence the genome of the variant. At that time, that was still difficult to do, but we did it. So we got a type of boring paper with a lot of potential explanation and so. But the interesting part is that we did a linkage map, and we obtained 16 groups, which is good because 16 is also the number of chromosomes. And there's one of them which is interesting, this one, because this is where you have GP9. Actually, PGM is also on that one. And this is quite interesting because for this chromosome, we had a bit more than 400 SNPs, and we found that 285 of them were completely linked. So there was no recombination into offspring produced by queens which were heterozygote for the small b and big B allele. So in this whole region, there was no recombination. And as I mentioned, this is a region which contained both GP9 and PGM3. So John Wan did some experiment, and he showed that there was an inversion, a large inversion, which is at least 10 megabases, which accounts for the lack of recombination. Actually, now we have done much more sequencing of 150 males, and we know that there are 3 inversions which overlap each other and which go up to the centromeres. And so that is a region which is about 12 megabases without any recombination. So to summarize, we have a species in which is extensive gene flow. There's no differentiation actually for 15 chromosomes, but 1 chromosome is really important. When you have 2 copies of the big B chromosome, you produce those large queens which will start new colonies on their own. When you have 1 copy of the small b haplotype, you have queens of intermediate size, and you have many such queens in colonies, but you can never have those queens being accepted in those colonies. And if you have 2 copies of the small b haplotype, this is not good because it's lethal, so that's the outcome of having 2 copies of that genotype. Now, this is very similar to something which is very common. Outside of Hymenoptera, you have another system which is very unusual. An idea? This is sex chromosomes. If you have 2 copies of this chromosome, you get this nice phenotype. If you have 1 copy of the Y chromosome, as you know, the Y chromosome is a bit degenerate, then you have this intermediate phenotype. If you will have 2 copies of the Y chromosome, then this is too much degenerate, you will get this type of phenotype. So this is very similar to sex chromosome. It means you have 2 phenotypes, which is due to 1 group of genes which are linked together, and there's no recombination in that case. So that can lead to degeneration, and here we have evidence of degeneration of the small b haplotype. We have a lot of transposable elements, some genes which are disrupted, and if you have 2 copies of this haplotype, then it's so lethal, just as if you will have 2 copies of the Y chromosome. So to conclude, we found that social organization defined Solenopsis invicta is regulated by a pair of social chromosomes. Actually, you only need 10% of heterozygote workers. We did some experiments, but you need 10% of workers with a heterozygote haplotype to make a shift from monogyny to polygyny, and then they will accept several queens, but only queens which have 1 copy of the small b haplotype. This region contained about 600 genes. So what would be interesting is to see what genes do what. And this is a difficult question because it behaves like a single genetic Mendelian element, and the light is coming, I'm happy. And the question is how this region evolved. So we know now it's this, we know that at screen version, we know that at about 500,000 years old, it evolved once, and then it was maintained in the 6 species, which has then evolved from a single ancestor. And we are now still studying the mechanism of the evolution of this region. So it's also this social chromosome is also very interesting because it underlies the presence of 2 social forms, and the properties of this region is very similar to that of sex chromosomes, and it's very interesting to see how those regions evolve because of the lack of recombination. And interestingly, there are many species which have variation in social organization, as I mentioned. We used to think, and that's what I used to think also, that this was a plastic phenotype, but we found in fact that it is not plastic. It's due to this supergene. And interestingly, one of my colleagues, also at the University of Lausanne, found that in another ants, Formica, difference in social organization is also due to a supergene. And it's not the same one. It's completely on different chromosome. It has a different origin. And interestingly, now he finds also that the same supergene is responsible for the presence of 2 types of social organization, not only in this Formica species, but in quite other Formica species. So I think supergenes, we heard also from the butterfly talk before, I think supergenes are very important, much more common than what people think, and they are responsible in many cases for the presence of alternative phenotype in animal, plant, and insect species. And there are an increasing number of such cases which are discovered, and I'm sure there will be many other cases which will be discovered in the near future. Now I'd like to thank you for your attention. I was so afraid that he would come to me.
Thank you for keeping your time and for this fascinating lecture. So are there questions? Yes.
So you made this parallel. You made this parallel with the sex chromosomes in mammals, for instance. But I see a big difference that, for instance, in mammals, you say Y individuals are lethal, but they're never produced, so there's no cost. But in those ants, small b individuals are produced, so there's a huge cost. So how such a system with a huge cost is maintained? And my second question related to that, you said that the small b allele is linked to transposable element and some kind of degeneration. But then how come that small b males, which are haploids, are viable?
So it's interesting. It depends on selection. So in that case, the small b males are viable. They produce less sperm than the big B males. And it's very interesting because it's probably due to the fact that there's strong selection at the haploid level on small b males because of the haplodiploid system of sex determination. And interestingly, if you look to sex chromosomes, in plants, there's expression of gene at the haploid level in the gametophyte, and so there's Y chromosome degenerate much more slowly in plants than in mammals. So in mammals, there's very little expression at the haploid level, so there's less selection, so it increases the likelihood to accumulate deleterious elements. And the fact that you have the haploid expression in ant males or in the plant, in the gametophyte, is very important to decrease the rate of degeneration. And also we found just now that there is a very low level of recombination, and this always has occurred ever since supergene evolved. And I think there is also some groups which tend to show that there can be sometimes recombination between sex chromosomes, also at a very low level, and that could be one mechanism which prevents speed of degeneration.
Yes?
How do the workers recognize the homozygous queen? Is it because it's bigger, or it is a different order?
It's an order. When I did the experiments, I noticed sometimes when I touch the big B queen, the forceps, workers will attack them. And even some workers which were attacking the big B queens were attacked by other workers. So we did experiments where we rubbed workers on the two types of queens. We introduced them in the mother colony. And we showed that when they were rubbed on the big B queens, most of them will be killed by workers. But there was no aggression when they were rubbed on the heterozygote queens. So it's ordered at the surface of the cuticle, which is responsible for the difference in behavior of workers. And we had identified some chemicals which are different. And we are currently working with a chemist in the US who synthesized some of those substances, and we'd like to find which one is really important. But we don't know yet.
Other questions? So I may have one. So you said there are some 160 genes in this region, and so this is the region where also the other gene that you had found initially mapped, right?
Yes.
EGM3. Okay.
Yes.
So how do you view these things here? Do you think there is a hierarchy among these genes? What type of functions are associated? Gene ontology associated with these genes?
This is difficult. So we know that many genes are differently expressed. Most of the genes which are differently expressed are actually in the region where there's no recombination. So there are actually 600 genes. Some of those genes...
because, for example, you have the one for the other binding protein,
yes.
but you have also certainly something with metabolism, because you said that the
There's
size
an
was smaller.
over-representation of genes which are differently expressed, which are linked to chemical signaling, which are in this region. We know that where there are the breakpoints of the inversions, of the 3 inversions, we know of 3 genes which are disrupted. So the transcripts are completely disrupted. And one of them could be responsible for the lethal effect. So I think we have, in another species, this gene may not be disrupted, so we may check if it's true or not. So our advantage is we don't have variants within a species, but with the 6 species, we have some variation, and now we are investigating what SNPs are fixed between species, and this could lead us to to have a fewer potential genes which are responsible for the phenotypic effects.
Thank you very much. So I think we move on.
Thank you.